So I'm going to talk to you this morning about new drugs and new treatment regimens. I'll start off by talking a bit about some of the new regimens that are being tested and then end up with some of the new molecules that are being developed. These, again, are my disclosures. So there's an extensive list. In another couple of weeks or months, it may no longer be possible to put all of the approved regimens on one slide, except fortunately a few drugs like didanosine and stavudine are coming off the market, and so may not have to be listed any longer. But as you can see, there are something like 30 entities, either as individual drugs or as fixed-dose combinations and single-tablet regimens that have been approved by the US FDA and by many other regulatory bodies around the world for treatment of people with HIV infection. So why do we need new drugs? Even though we have really terrific drugs, there are still some regimens that have side effects or we discover some adverse effects of drugs the longer we have experience with them. There are potential long-term toxicities of some of the current treatments that we use. There's always the concern about the development of resistance over the longer term. As effective as our drugs are, and as much as I said yesterday that resistance has become an infrequent problem in the resource-rich world, we know that there's generation of resistance to protease inhibitors when there's not careful virologic monitoring, certainly in South Africa, where I've been working, and I think we can't be too content that we've solved the resistance problem and we won't need new drugs in the future. And we really need to work on less frequent dosing. Daily dosing is terrific. Yearly dosing would be spectacular. And so that has to be a goal to keep in mind. So let me start by talking about TAF, a drug which is approved now in many places, but is still more or less a new drug. As you know, tenofovir is the active drug. Tenofovir is a phosphonate, so it already comes with one phosphate. The active drug is tenofovir diphosphate. And up until recently, the formulation of tenofovir, which by itself has poor bioavailability, was tenofovir disoproxyl fumarate. And TDF gets absorbed in the gut and circulates in the plasma, where it's converted to tenofovir on absorption and then gets into the cell. So you need relatively high concentrations of tenofovir in the plasma to be able to get sufficient cellular concentrations. On the other hand, TAF, a newer formulation, the Alafenamide formulation of tenofovir, is not metabolized in the gut or plasma, and is concentrated in lymphoid cells and in hepatocytes. And there it is, the alafenamide is cleaved by cathepsin A to liberate tenofovir, which is then phosphorylated to the active drug. This allows much lower doses of TAF to be administered and much lower plasma concentrations of tenofovir as a consequence, and still achieve dramatically higher intracellular concentrations of the active compound. And so here you can see from one of the phase 2 studies that when administered as TAF, you actually get 6.5 times higher intracellular concentrations of the active drug compared to the TDF formulation, even though the plasma concentrations of tenofovir are either the same or are lower. Here, the actual concentrations shown here. So you have basically 1/7 the plasma concentration, and nevertheless 6 or 7 times higher intracellular concentrations. So how does TAF work as compared to TDF? Well, just as well, as you might expect. So these are data, again, from my colleague Paul Sachs, comparing ECF-TAF to ECF with TDF.